Hello, everyone. Welcome back to my video tutorials for single-cell RNA sequencing data analysis. In this video, I'm going to show you how to perform data integration with SCTransform-normalized dataset in Seurat v5. So first we can load all the packages Seurat, Tidyverse, Patchwork, and SCTransform, we can set the set. In my previous video tutorial, we loaded 3 GSE datasets for human non-single-cell RNA sequencing from healthy donors, and we created an object and performed the data integration. So today we can use the same dataset for the demonstration. We can merge the 3 datasets as object SCT. Let's merge the dataset. Then we don't need the 3 datasets anymore. We can remove it. So now we can have a look at the metadata for the object SCT. Let's run. You can see we have the barcodes as the row names. We have the GSE dataset name in the origin ident column. Then we have the uncount RNA, M-feature RNA, and the percentage mitochondrial DNA. As I demonstrated the data analysis in Seurat v5, so we can have a look at the C information for the RNA. If we run, you can see it is a C file, Seurat object. So normally, if we do the analysis, we need to run data normalization, find the variable features, and scale the data. The SCTransform is an improved method to normalize the data. If you run SCTransform, it automatically runs 3 steps to normalize the data, find the variable features, and scale the data. When we run SCTransform, we can also remove confounding source variations. For example, here we can remove the mitochondrial DNA percentage variations. So now we can just run SCTransform to normalize the data, find the variable features, and scale the data. Before we do that, we can have a look at the object. You can see in the C, we only have the RNA, and we have 3 layers from 3 different datasets. So now we can run SCTransform. Let's run. Because we are doing 3 steps analysis at the same time, so this step will take some time to finish. Okay, I finished the SCTransform analysis. We can have a look at the data structure for the object again. If we click the object, you can see now in the assays, we have 2 assays. One is RNA, the other one is SCT. So, SCTransform analysis created another assay, SCT assay. In the SCT data slot, we have the SCT model list. We have 3 data information here. We also have the counts data here. Let's normalize the data by the SCTransform analysis. And also we have another data slot here. You can see we have the scaled data and the variable features. So following the SCTransform analysis, the default assay will be SCT. If we run default assay for the object, let's run, you can see it is SCT. And also we can have a look at the metadata. You can see now we added the uncount SCT and the in-feature SCT in the metadata. So now we can go to next step of analysis. Next step will be run PCA. Let's run it now. So following PCA analysis, we can run the elbow plot to identify the best pieces for data integration and downstream analysis. Let's run the elbow plot. You can see in the elbow plot, the elbow shape is somewhere here. So if we choose PC from 1 to 20 will be okay. For today's demonstration, I'm going to use 30. So any number after like 20 will be fine. So now we can run the same method for data integration, the RPCA data integration method. Because we normalize the data using SCTransform, here you need to set the normalization method as SCT. Then we can perform the data integration. Let's run. So after data integration, we can find the neighbors. As I mentioned, we are going to use PC1 to PC30. Let's run. So next, we can find the clusters. I'm going to use lower resolution because this will be easier for us later to demonstrate the find the cluster function. Okay, we can run find clusters, and also we named the cluster name as RPCA clusters. Let's run. So you can see with resolution 0.2, we have 11 cell clusters from this dataset. Now we can run UMap. And we give a reduction name as UMap RPCA. So we finish the analysis. Now we can have a look at the metadata again. Let's run. You can see now we added the cluster number for each cell in the metadata. You can see the RPCA clusters and also the same cluster number for each cell as Seurat clusters. So let's go back to look at the cell clusters using the dim plot function. First, we can plot the cells using dim plot and then group the cells by the RPCA clusters. Let's run the dim plot. You can see we generated the UMap. The UMap is UMap RPCA. Then we have 11 cell clusters here. We can also run it by origin ident. Then we can show the cells from different datasets. Let's run. You can see now cells from GSE 128033 are in red. This dataset don't have a lot of cells. Then cells from GSE 128169 are in green and GSE 132771 in blue. You can see we use the SCTransform to normalize the data, then integrated the cells by RPCA method, and the cells from different datasets were integrated very well in their cell clusters. If you want, you can also use the split by function to see the cells by different datasets we can run. You can see now the UMap shows cells from different datasets. So after analysis, you can use mark genes to identify the cell types. Here I just use for classic mark genes for the non-cell types. Let's run the feature plot. and we can zoom in. You can see collagen 1A2 labels mesenchymal cells, EPCAM identifies epithelial cells, CLDN5 identifies endothelial cells, and PTPRC labels immune cells. This is the feature plot. You can also run volume plot. We can use PTPRC as an example. Let's run volume plot for PTPRC. You can see cluster 0, 1, and 2 express high-level PTPRC. So cells in cluster 0, 1, and 2 are immune cells. So the last demonstration I want to show you is find the max function. Because we normalize the data using SCTransform, and the normalized data were saved in the SCT data slot. So if you want to use the find the markers function, first you need to run the prepare SCT find the max function for the object. Let's run. You can see here it found 3 SCT models. I showed you in the model list. There are 3 models for 3 different datasets. So after this step, now you can find the markers. For example, we can use cluster 1 as an example to identify mark genes for cluster 1. We know cells in cluster 1 are immune cells. We can run the find the markers function. Let's run. You can see we generated the data frame to identify mark genes for cells in cluster 1. Let's click the data frame. We can reorganize the log2 folder changes. You can see most genes are expressed by immune cells. For example, CD3 is a mark gene for T cells. Okay, that's today's demonstration to show you how to perform data integration with the SCTransform-normalized dataset.